Good morning all. Welcome to Sri Education and Inclusive Learning. Here I am Sreejisha. I am a postgraduate in microbiology and also holding B.Ed, and I am a teacher. So today I am having my new venture with a new online platform with an intention to have free education for students, those who are in need of. Okay, so let's start with the new topic. I introduced a new topic, the cell, the basic unit of life. In this I had included the basic concepts regarding cell, some of the discoveries, cell theory, cell biology, and some of the terms to be memorized from the concept. Okay, let's move on to the next slide. Okay, coming to the topic, that is cell is the basic unit of all organisms. So you all know all organisms, we itself, are composed of different types of cells. And the cell is the structural and functional unit of life. And the study of this structure and composition of cells with the aid of microscope and modern technologies is known as cytology. Cell biology, it is the branch of biology which contains the biochemical techniques such as exfoliation of the cell, cell culturing, and biopsies, and regarding all the studies regarding the cell which is collected, where from it is collected, and also it is concerned with the organization and function of the particular cell. Cell theory was proposed by Theodor Schwann in 1839, and he postulated that all living organisms are made up of one or more cells, and the cell is the basic unit of the structure and the function. In 1665, Robert Hooke was the scientist who first used the term cell. In his publication Micrographia, he illustrated the smallest complete parts of an organism, which he called cells. His theory that all organisms are made up of cells, which are the basic structural units of life. He found the cell in cork tissues with the help of his compound microscope invented by himself. Robert Brown, in 1831, he discovered the nucleus in orchid roots. In 1831, while investigating the fertilization mechanism of plants in the Orchidaceae and Acalyphidaceae families, he noted that the existence of a structure within the cell of orchids, as well as many other plants, that he termed the nucleus of the cell. While the discoveries were going on regarding the cell, the term totipotency was coined by Gottlieb Herberland. Cells are totipotent. According to him, the ability of a single cell to divide and produce all the differentiated cells in an organism, including extra embryonic tissues, is known as totipotency. Totipotent cells formed during sexual and asexual reproduction includes spores and zygotes. Rudolf Virchow in 1855 proposed that cell arise from pre-existing cells. He published a statement based on his observation Hominis cellulae e cellula, which means that all cells arise from the pre-existing cells. Virchow used a theory that all cells arise from the pre-existing cells to lay out the groundwork for cellular pathology, or the study of disease at the cellular level. Many of his discoveries include finding cells in bone, connective tissues, and describing substances such as myelin. He was the first person to recognize leukemia. Antony Phillips van Leeuwenhoek, he was the person who discovered the living cell, that is the living cell, the bacteria. Most important discovery was the existence of a single cell organism. While using a microscope to examine the pond water in 1674, he observed dozens of protists, which he called animalcules, as well as Spirogyra or green algae. These animalcules were the bacteria. Volume determines the amount of chemical activity of cells per unit time, while surface area determines the amount of absorption and release of the products in and out the cell. The greater the surface area to volume ratio of a cell, the easier it is for the cell to get rid of waste and take in essential materials such as oxygen and nutrients. Now coming to the types of cells, basically there are two types of cells, prokaryotic cell and eukaryotic cell, based on the presence of nucleus inside the cytoplasm. So coming to prokaryotic cell, it is having a single membrane and are haploid cells which have respiratory enzymes associated with the plasma membrane. Respiratory enzymes means these are the enzymes such as oxidase, which catalyze the transfer of electrons from its substrate to the molecular oxygen during cellular respiration. In prokaryotic cells, it is having circular DNA without any proteins, forming a single chromosome and lying in direct contact with the cytoplasm and 70 years ribosomes. So in the prokaryotic cell, we can see there won't be a definite nucleus inside. The genetic material DNA will be having direct contact with the cytoplasm. Now coming to eukaryotic cells, they are having double membrane and are diploid cells which have plasma membrane devoid of any respiratory enzymes. These are having linear DNA which is associated with the proteins, forming 2 to many chromosomes enclosed by a nuclear envelope. So there is the difference coming. This eukaryotic cell is having a definite nucleus. You can see it in the cytoplasm. And inside the cell there are ATS ribosomes with many membrane bound organelles. Inclusions are present. And eukaryotic cell, for example, animals, plants, fungi, protists, you can see eukaryotic cells. Next, coming to fluid mosaic model by Singer and Nicholson. It is the biomembrane consisting of classified phospholipid bilayer with embedded proteins. This model explains the structure of plasma membrane of animal cells as a mosaic of components such as phospholipids, proteins, cholesterol, and carbohydrates. The peripheral membrane proteins are also loosely held on the surface of the bilayer. Now coming to the anatomy of a cell. The cell consists of different membrane-bound organelles, inclusions, cytoplasm inside. The anatomy of the animal cell is different from that of plant cell. Plant cell is having a thick cell wall surrounding the cell, and here in the animal cell you can see the cell wall is little thinner. Also, this cell wall is giving the structure and shapes and support for the cell. Also, you can see the cell membrane is there. Cytoplasm, the blue color viscous liquid present inside is called cytoplasm, and you can see there is a definite nucleus with a nuclear envelope and the nucleolus inside. The nucleolus consists of the chromosome, which is consisting of genes, and here the genetic material DNA is present. And also in the cell cytoplasm you can see lysosomes, mitochondria, the powerhouse of the cell, centrosomes, which are the primary microtubules, Golgi bodies are there. The green color thing is the endoplasmic reticulum, and there is a definite nucleus compared to the prokaryotic cell. The eukaryotic cell will be having a definite nucleus, and that's all about the cell. So we'll go in detail regarding the things such as cytoplasm, what is cell wall. So the cell consists of cell wall, cytoplasm, and all what we see. So coming first to cell wall, the plant cells are enclosed in cell wall, and also animal cells are also enclosed in cell wall. But the plant cell composed of cellulose and other substances as chitin, pectin, mannose, and the nucleus. A cell wall is a structural layer which is surrounding some types of cells just outside the cell membrane. It can be flexible, sometimes rigid also. It provides the cell with both structure, support and protection, and also acts as a filtering mechanism. In bacteria, that is in prokaryotic cells, the cell wall is composed of peptidoglycan. Cytoplasm, it is the thick solution which is transparent that fills inside each cell and is enclosed by a cell membrane. It is mainly composed of some of the waters, salts and proteins, and also cell organelles and cell inclusions can be seen inside this. Coming to the nucleus of the cell, it is the main center part which controls all the cell activities. It has a nuclear membrane inside which the chromosomes bear the genetic material DNA in the genes. There are special types of chromosomes such as Lamb-Rush chromosomes and polytene chromosomes. Okay, now coming to the next one, that is mitochondria. It is the powerhouse of a cell. It is having a double membrane bounded around it and is also enclosed a fluid matrix. Oxysomes are structures which present inside the cristae of the mitochondria, which are used for ATP synthesis. That's why it is called as powerhouse. ATP is synthesized and stored in it for the cell. Now coming to chloroplast. Chloroplasts are the organelles that conduct photosynthesis. These are the plastids that contain the green pigment chlorophyll, which are covered by the double membrane, and they are the sites of photosynthesis, where the photosynthetic pigment chlorophyll captures the energy from sunlight and it converts it into the energy, and this is stored in the energy storage molecules ATP and NADPH while freeing the oxygen from water in plant and algal cells. Coming to endoplasmic reticulum, that is ER, it was discovered by Porter and Thomson. It is a three-dimensional structure. The endoplasmic reticulum, it is in essence, it is the transportation system of the eukaryotic cell and has many other important functions such as protein folding. It is a type of organelle made up of two subunits, that is rough endoplasmic reticulum and smooth endoplasmic reticulum. The rough endoplasmic reticulums are having the ribosomes on its surface and it is called as rough endoplasmic reticulum. The surface of the smooth endoplasmic reticulum will be more smoother than the RER. So we said about the ribosomes which are present on the surface of endoplasmic reticulum. Also the ribosomes are present in the cytoplasm. You can see the free floating ribosomes in the cytoplasm. It was first discovered by Robinson and Brown in plant cells and Pallade in animal cells. It is a membranous ribonucleic proteinic protoplasmic structures with a size of 150 to 200 amps, which are the sites of protein synthesis. Now next coming to Golgi apparatus or Golgi bodies. It was first observed by George in 1867 and named by Camillo Golgi in 1898. It is a complex protoplasmic structure made up of smooth membrane sacules or cisternae, a network of tubules with vesicles and vacuoles, which take part in the membrane transformations. And saying about the cisternae, it is a disc-shaped sacs in the Golgi apparatus. And the vacuoles, food vacuoles, these are the cytoplasmic vacuoles. There are many vacuoles, contractile vacuoles, food vacuoles, sub vacuoles, etc. Coming to microtubules, these are the unbranched hollow submicroscopic tubules, the polymers of protein tubulin, and that form the part of the cytoskeleton and provide the structure, shape, etc. for the eukaryotic cells. Microtubules can grow as long as 15 micrometers and are highly dynamic. The outer diameter of the microtubule is between 23 to 27 nanometer, while the inner diameter is between 11 and 15 nanometer. Centrosomes are the primary microtubule in animal cell, which regulates cell mobility, adhesion and polarity to the cell. Now coming to microfilaments, they are in 8 nanometer in diameter and formed of actin or myosin, either scattered or arranged in a parallel rows, help in the managing the stage of the cell. Okay, coming to cytoplasmic vacuoles, there are maybe sub vacuoles are there, contractile vacuoles are there, food vacuoles, ASI vacuoles are there. Vacuole is a membrane-bound cell organelle. In animal cells, the vacuoles are generally small and help in sequester the waste products out of the cell. In plant cells, the vacuoles help maintaining water balance. Sometimes a single vacuole can take up most of the interior space of a plant cell. These are essentially the enclosed compartments which will be filled with water containing inorganic and organic molecules, including the enzymes in solutions. Coming to cilia and flagella, these are the slender hair-like structures that protrude from many types of cells. In many cells, the cilia acts as the fundamental unit of motion, saving in the mobility of the cell as a key organelle that convert chemical energy into mechanical work in the form of an oscillatory beating motion. It is in 9+2 arrangements in many cells. Coming to lysosomes, these are membrane-bound organelles which are involved in the degrading and recycling the cellular waste. In cellular signaling and energy metabolism inside the cell. So these are the things in the cell which I am concluding the concept here. And now going to the terms to be memorized. We can see some of the terms which have to be memorized, like totipotency. As I explained, it is the ability of the cell or the potential of the cell to give rise to a new individual or the new cell. This potential of a cell is known as the totipotency. Stem cells are the undifferentiated cells, which are like the meristematic cells in plant cells, which continue in dividing, help in the organism's growth. Coming to organelles, these are the living structures of the cells, live structures of the cells with having definite functions, like mitochondria, endoplasmic reticulum, Golgi apparatus, etc. Now coming to inclusions, these are the lifeless structures in the cell which may or may not have definite functions, like vacuoles. In many plant cells and all, there are vacuoles which will be collecting only the waste inside the cell. Coming to the other terms, cytoplasm, hyaloplasm and nucleoplasm. First term, cytoplasm, as we already seen, it is the thick solution which is transparent that fills inside each cell and is enclosed by a cell membrane. It is mainly composed of some of the waters, salts and proteins, and also cell organelles and cell inclusions can be seen inside this. All the organelles in eukaryotic cells, such as the nucleus, endoplasmic reticulum, mitochondria, are located inside the cytoplasm. From the term itself, we can be clear from this word. Cyto means inside the cell. Now next, coming to hyaloplasm, it is the clear homogeneous ground substance of the cytoplasm. It is also known as cytoplasmic matrix, which is present in between the foldings of ribosomes and endoplasmic reticulum and all. These are known as hyaloplasm. So coming to the next one, that is nucleoplasm. From the word it is clear, nucleoplasm means inside the nucleus, which is plasm means the fluid. So it is the fluid which is present inside the nucleus. That is a clear, transparent, homogeneous fluid which is inside the nucleus, covered by a nuclear envelope and nuclear membrane. So from this, here you have to remember, cytoplasm means the liquid inside the cell, and nucleoplasm is the nucleus which is inside the cell, which is having the liquid inside the nucleus. that is with the liquid inside the nucleus is called nucleoplasm. By hearing the word itself, you have to identify cyto means inside the cell, and nucleo means inside the nucleus. Okay, so thus from here I am concluding my concept. So coming videos we can see with other extensions of this thing, the cell. Hope you all enjoyed the video. I will be coming with other concepts and extended concepts of cell. In that we can see about the detailed things, some of the questions which will be very important. 5 mark or 3 mark questions will be very short answer and briefing questions we can see. So thank you all for watching this video and help me in going beyond this venture. Thank you all. Please like, share and subscribe for the same. Hope you will all be with me to excel more in this one. Okay, thank you so much.